Hi, it's Mr. Rico, and welcome to IB Biology. So in today's video, we're going to go over carbohydrates and lipids, B1.1. We're going to start with carbs, and then we're going to finish with the lipids and oils. The main learning goal of this topic is to examine the chemical structure of these compounds and relate to their biological function. All right, so let's go. First of all, our brains, they are wired for carbohydrates. We seek we seek for this. We want this. This is tasteful for us, and we are wired to search for them because in the past, carbohydrates meant survival, meant energy, and a source of energy for our bodies. Okay, and typically, at least these ones here, they're pretty healthy. If you eat them, you're doing good. Okay, but the problem today is that it's very, very easy to overeat these substances, the carbohydrates. Why? Because especially with sugarcane, it's so cheap to produce sugar that we can easily add to any source of industrialized food. For example, these candies, sweets, soda, Nutella, donuts, whatever. It's so easy to add them, and it's easy to, let's say, eat some of them and explode your daily intake of simple or refined sugars. Okay, good. Now let's look at their structure. The first thing is they are carbon-carbon compounds, which means that they have several carbons on each structure, and of course linked with hydrogens. We're going to see that the carbohydrates have a lot of oxygen as well. Okay, but this is their basic structure. They have a carbon-carbon bonding also with hydrogens and oxygens. This image here in the top is an image of glycogen. Okay, so we can see the structure of this chemical is highly branched and complex. We have a glucose as well. We're going to talk a lot about this. This is forming a ring, and we have a lot of oxygen. So this molecule is also a polar, have poles, polarity in the molecule. And here down we have a typical saturated fatty acid. We can see the saturations in the bonds. So we have several bonds, single bonds between the carbons. And in each one of these bonds, you have a lot of energy that can be oxidated by oxidative reactions in the mitochondria. And we also have our carboxyl group here in the end. Okay, so we can clearly see some differences between them. Okay, this is just one example of how this sugar is made, how the glucose. Okay, we can demonstrate in a linear form with the 6 carbons. Okay, or we can demonstrate in a circular form as it's shown here in the right. Good. So carbohydrates, we also call them sugars. We also call them saccharides. You might see this terminology, the same terminology for all of them, but basically they mean the same thing. Okay, sugar, saccharide, carbohydrate. One is more formal or more informal, but it's the same thing. Good. So first, let's start with the building blocks, the elements, the key elements of all carbs. What we call mono-1 saccharides, one sugar. Okay, so monosaccharides, they're basically made of one unit, one building block of sugars. So we have three building blocks. We have the glucose, we have the galactose, and we have the fructose. Slight differences between them, but they end up having differences in what they build. Okay, they're soluble typically, and glucose is the one. Okay, is the molecule in terms of carbohydrates. Mainly from glucose, we build several other polysaccharides. Okay, so monosaccharides. If you combine two units, two monosaccharides, we have D saccharides. So D2 saccharides sugars. So two sugars together. Okay, let's check some examples. Here, for example, we have two monosaccharides in a condensation reaction. So we have one hydroxyl group combining with another atom of hydrogen here in this reaction, and we end up condensing one water molecule, and we form this bond between the two units. So we form disaccharide. Examples of disaccharides: maltose, lactose, and also sucrose, the table sugar that we add to stuff. If we continue on this line, on this thinking, we're going to have the oligo. Oligo is just a few, okay, some units or some dozens. Saccharides, so some sugars connected, for example, 5, 10, or some dozens. Okay, they're not that common. They're not that important, especially for IB biology, but we just talk about them. And then we go to the important ones, the polysaccharides. So poly in Greek means many, a lot. Saccharides, sugars. So a lot of sugars, a lot of carbohydrates connected, linked to one another. Okay. The main examples of polysaccharides. First, we have cellulose, the fibers of plants. Whenever you see a tree, a plant, or even your vegetables, we're talking about cellulose. Tons and tons of glucose connected together, forming these bonds. We also have amylose, that is a form of starch, along with amylopectin, that is another form of starch. And we can see that we have several units, thousands of units of sugars connected. Depending on their branching framework, you can build one type of starch or another type of starch. And then we finally have the glycogen. Okay, so the glycogen is a substance that we have in our bodies that we store, and we can break down to generate glucose, to generate glycol, especially in our liver and on our muscles, depending on the physical exercise that we are doing. So these are the most famous polysaccharides that we know and that we talk in biology. And of course, they're of great importance in nutrition. Here again, we have some examples. We have the starch amylose and amylopectin. Depending on the chain, if it's branched or unbranched, we're going to make different types. We're going to separate their types of starches. If we look at a glycogen, interesting. So this I showed before, and this central view, we have a protein in the center, and we can see how many glucose units are linked and are branching from this glycogen. Once you start to need glucose in your bloodstream, your cells can come here, and with specific enzymes, they can break down these bonds, and they release in the bloodstream each one of these units. So then you can use across your body and in your mitochondria, right? Next, polysaccharides has structural components. Cellulose, fiber, it's all cellulose. It's all carbohydrates. And even wood, whenever you see wood in a furniture, in a house, it's basically cellulose. It's basically glucose. So we form these molecules, we form these fibers with billions and billions of glucose units, and we also form these cross bonds, and we link them all together, forming fibers that compact on one another. Depending on the density, you're going to have different strands of this wood. And finally, they make up the cell walls of plants. And the part of plants that can accumulate more can be, and that can have more density, are the trunks, the wood. Interesting. Okay, so we've seen so far, we have some chemical patterns here. We can see that the carbon, hydrates, water, hydration. We can see that the carbohydrates, they follow a typical pattern, a typical chemical ratio. All right, and this ratio is what I'm showing here. C carbon, water. So carbon, water, one carbon to water. So we're gonna see that the chemical formulas of carbohydrates, they always follow the CH2O chemical ratio, right? So if we check monosaccharides, like the galactose, it's C6H12O6. If we continue to the disaccharides, multiply by 2, it's just combining 2. For example, lactose, galactose, and glucose. It's C12H24O12. So on and on with maltose and sucrose as well. And if we get the polysaccharides, for example, cornstarch or pasta or glycogen, they're going to be massive. They're going to be huge molecules, but they're going to follow the same chemical ratio. Okay? All right. Once we build the carbohydrates, especially plants build this by photosynthesis. If we build them, if you eat And if you digest, how do we break? If the anabolism of carbohydrates is called condensation, okay, the catabolism, the breaking down, is called hydrolysis. So you use a molecule of water, okay, and you hydrolyze, you break down this molecule. You insert one molecule of water as you give one hydroxyl and one hydrogen atom here. Once you have this hydrolysis, you separate from disaccharides to two monosaccharides being released in the reaction. The condensation is the exact opposite as we saw before. So the condensation is when you condense, is when you form one water molecule, okay? Okay, this is an interesting activity with some models, with some kits, with some models to build some molecules. This is an interesting practice. If you have the model, this is an interesting activity for us to summarize these chemicals. Now let's move to lipids. Now let's jump into the oils and fats. Again, there is no additional higher level for B1.1, so it's all standard level. Good. So what are oils? Okay, we can see the effect of oils, for example, wax or oils like these, in these feathers, because they repel water. They don't mix. There's no solution between, there's no solvation between water and oils. We're going to see why. What is the chemical explanation for this event? Here are the classes of lipids. We have waxes, we have triglycerides, we have three molecules. We also have the phospholipids that are very important for the membrane structure and membrane transport. They have a phosphorus head and a lipid tail. And the steroids, the hormones that we have and that we use in our body. Properties and functions of lipids. So here I have this diagram showing hydrocarbons. So hydrocarbons, they show perfectly what it is. It's typically they are long chains of carbons and hydrogens, carbons and hydrogens, carbons and hydrogens. This is why hydrocarbons. Okay, and if they are saturated, they're going to be called saturated fats. Okay, so saturated, it means that they are saturated. They only have single bonds, single covalent bonds between the carbon and the next carbon. If they are unsaturated, they have at least one unsaturation on its structure, and they can be cis or trans. We're going to see how it works. Okay. So the fats, they're typically found in animals. Okay, so animals like animal fats like butter or found in meat. They're saturated, they have straight chains, and they're solid at room temperature. Why they're solid at room temperature? Because they can be compact to one another and they get back together and be solid. Okay? Plants, on the other hand, they tend to build unsaturated fats like olive oil, and they have a kinky tail. As they have the unsaturation, they end up being like kinky with their chemical tail, and they are liquid at room temperature because they can be all super compact and connected to one another at room temperature. So they're liquid at room temperature. Unless if you cool down the mixture, maybe you can turn them into a solid. Okay, we also have the triglycerides and phospholipids. Okay, so the triglycerides, they have a glycerol group and these long chains of carbon to carbon. Okay, so there's why triglycerides, we have three of them. They're very important, they're very common in our body. We have several uses for them. And just recalling this saturated and unsaturated, again, saturated, straight, all linear. Unsaturated, we have at least one. If we have one, monounsaturation. If we have many, polyunsaturation. Typically, the unsaturated fats, they tend to be healthier than the saturated counterparts. Okay, the most obvious and known example of a saturated fat is butter that comes from milk, and unsaturated, as we see here, with the kinky tail, liquid at room temperature, that is pretty healthy, is olive oil. Good. So here we can think about it. Are these sources good or bad? Okay, it depends, but you might think about it like eggs, cheese, meat, bacon, chips, milk. We have several sources of fats. So think about it. Are they saturated, unsaturated? Are they healthy, unhealthy? Here, the same thing. Are they saturated, unsaturated, polyunsaturated? Are they healthy? Yes or no? This is a good question that we can pose to our lives and also to our exams, because we have several questions that can cover this, can cover the influence on humans and on health. Okay, talking about phospholipids, we have the formation of bilayers. Okay, as we have a hydrophilic region and a hydrophobic tail, they can form bilayers by attracting the hydrophilic to hydrophilic and hydrophobic to hydrophobic. So they eventually form these circles, or they can form a bilayer on water. This is the typical membrane of cells. Okay, again with a phosphate, polar head, and the saturated tail. Okay, we're gonna also study this on membrane structure. All right. And triglycerides, they are widely used, especially mammals and other animals, for storage of energy and thermal insulation. For example, polar bears, in whales. Okay. And finally, non-polar steroids, they can also pass through the phospholipid bilayer because they're basically non-polar and made of carbon and hydrogen mostly, and they're also the basis of hormones. So the steroid hormones, they also come from fats. They also come from lipids. They have a very similar structure when we compare. That's it for today. We covered carbohydrates and lipids, and I hope this was helpful.